We completed the first year syllabus still genetics. Today we discuss second year syllabus first chapters Introduction to Plant Kingdom, Spirogyra and Rhizopus. Introduction to Plant Kingdom. Eichler proposed the first phylogenetic system of classification. In that he classified the plants into two major kingdoms, cryptogams and phanerogams. Cryptogams are non-flowering plants. Here these are again three classes: Thallophyta, Bryophyta, Pteridophyta. These are subdivisions in this or divisions in this. Thallophyta, plant body is a thallus, undifferentiated plant body without root, stem and leaf, with autotrophic thallophytes, algae, heterotrophic thallophytes, fungi. Then bryophytes are amphibians of plant kingdom because they completely depend on water for fertilization. Pteridophytes are first group of vascular plants, so they are also called vascular cryptogams. Then we discuss phanerogams, flowering plants. In that, gymnosperms, their only seeds are formed, fruits are not produced. Why? Because ovary is absent, only ovules are present. Then comes angiosperms, where ovaries and fruits are present. Different types of categories in that. Then Spirogyra, algal example. Spirogyra discovered by Link. It is an autotrophic thallophyte, grows in freshwater habitats, popularly called pond silk, water silk, pond scum. Structure of Spirogyra filament, structure of cell. Asexual vegetative reproduction by fragmentation, asexual reproduction by echinids and eplanospores, sexual reproduction by conjugation with scalariform conjugation, lateral conjugation. If two filaments involve scalariform conjugation, if two cells of the same filament involve lateral conjugation, formation of zygospore, haplontic life cycle and other things. Then Rhizopus, heterotrophic thallophyte, grows on bread, hence it is bread mold, pin mold, black mold, discovered by Ehrenberg, represented by 30 species. Well known species is Rhizopus stolonifer. Structure of mycelium, structure of hyphae, stolons, rhizoids, sporangiophores. Stolons and rhizoids formed during vegetative growth. Sporangiophores are formed during reproductive growth. Then how asexual reproduction takes place by eplanospores, how sexual reproduction takes place by gametangial contact. In the gametangial contact, fusion of nuclei, disintegration of unpaired nuclei, then formation of zygospore, reduction, division in the zygospore, formation of promycelium or germ tube and germ sporangium. The basic difference between germ sporangium and sporangiosporangium is in sporangiosporangium no question of formation of columella. In germ sporangium, columella is not formed. In sporangiosporangium, columella is produced. Then spore formation, meiospores, the only structures in the total life history of Rhizopus which are uninucleate at least for a short period of time are meiospores, are the spores formed in the zygosporangium, that is zygospores. So all these things we discuss in the form of important questions. Now first we take first question. Which of the following is true with regard to thallophytes? So this is a question regarding thallophytes. Which of the truth means they are non-embryophytes? Definitely they are non-embryophytes, because in thallophytes, zygote diploid, instead of mitosis it undergoes meiosis and produces haploid structures. Whenever zygote undergoes mitosis, then it produces embryo. Whenever zygote undergoes meiosis, it does not produces embryo. So all thallophytes are non-embryophytes. Why? Because they don't produce embryo, because zygote instead of showing mitosis shows meiosis. So these are non-embryophytes. Next, second, most of them have multicellular and non-jacketed gametangia. Multicellular, non-gametangia, non-jacketed. Multicellular or unicellular you have to write. Then they are atracheophytes. Atracheophytes means absolutely they do not possess any vascular tissues. They are atracheophytes is correct. They show haplontic type of life cycle. It is also correct. Haplontic means in the total life cycle, zygospore is the only diploid stage. All the other stages are haploid. Hence it is called haplontic. So according to this question, the question is, which of the following is true with regard to thallophytes? Is they are non-embryophytes is correct. Most of them have multicellular and non-jacketed gametangia is wrong. All of them have multicellular non-jacketed gametangia. They are atracheophytes, it is correct. They show haplontic type of life cycle is correct. So answer is 1, 2 and 4. For the first question, Which of the following is true with regard to thallophytes? They are non-embryophytes, they are atracheophytes, and they show haplontic type of life cycle are the best characters regarding thallophytes. Which of the following characters are not related to pteridophytes? Not related to pteridophytes. Pteridophytes are the first group of plants. Thallophytes are gametophytic generation is dominant. Bryophytes, gametophytic generation is dominant. For the first time in pteridophytes, sporophytic generation is dominant. So whenever sporophytic generation is dominant, the plant body shows root, stem and leaf perfectly. So which of the following characters are not related to pteridophyta? Means plant body is a gametophyte is wrong. Plant body is not a gametophyte, plant body is a sporophyte, diploid sporophyte. Siphonogamous type of oogamy. Pteridophytes does not show siphonogamous type of oogamy. What is siphonoogamy? Movement of male gametes from one place to other place with the help of a pollen tube is called siphonogamy. Pollen tube formation starts from gymnosperms. Gymnosperms and angiosperms show siphonogamy. Pteridophytes absolutely does not show siphonogamy. So siphonogamy is siphonogamous type of oogamy is wrong. Direct pollination, no question of pollination in pteridophytes. Microspores divides and produces prothallus. Sexual reproductive structures are found in prothallus or gametophyte. Reproduction takes place. Sessile sex organs. Sessile sex organs is correct because all pteridophytes, the special feature is reproductive structures, antheridium and archegonium are sessile. They do not possess any stalk. So which of the following characters are not related to pteridophytes is sessile sex organs are related to pteridophytes. Not related, plant body is not a gametophyte. Plant body is a sporophyte, so that one is one. Siphonogamous type of oogamy, no question of formation of pollen tube in pteridophytes, so siphonogamous type of oogamy. Direct pollination is found in gymnosperms, not in pteridophytes. So 1, 2, 3 are not the characters which are related to pteridophytes. Which of the is true regarding to bryophytes? Regarding to bryophytes. Bryophytes are the first successful land plants without vascular tissues. First time in the plant's embryo is produced because zygote has undergone mitosis. These are the embryophytes. Embryophytes ultimately become bryophytes. Now they are embryophytes is correct because for the first time embryo is formed. They are atracheophytes is correct. Atracheophytes is correct because bryophytes, the total plant body is filled with parenchyma. No question of vascular tissues, hence they are atracheophytes. Then life cycle is haplodiplontic with heteromorphic alternation of generations is correct. Haplodiplontic because majority of the stages are haploid. Only few stages are diploid. That's why this is the only group. Bryophyta is the only group which shows haplodiplontic. Why? Because algae shows haplontic, fungi shows haplontic, pteridophytes, gymnosperms, angiosperms show diplohaplontic. Haplodiplontic cycle is found only in bryophytes. That's why the life cycle is haplodiplontic and heteromorphic alternation of generations. Heteromorphic alternation of generations means sporophyte and gametophyte are entirely different morphologically and physiologically, but they perfectly alternate with each other. Perfectly alternate with each other is alternative heteromorphic alternation of generations. They grow in moist, shady places. Bryophytes always prefer to grow in moist, shady places. Why? Because they completely depend on water for fertilization. That's why they are called amphibians of plant kingdom. So they are embryophytes, they are atracheophytes. Life cycle is haplodiplontic with heteromorphic alternation of generations. They grow in moist, shady places. All four characters are correct for a bryophyte. So 1, 2, 3, 4 will be answer. That's why the answer will be 4. Now this is an assertion reasoning question. Endosperm in gymnosperm is haploid. Endosperm in gymnosperms is definitely haploid. It is a pre-fertilizer product, female gametophytic tissue formed from haploid megaspore. In gymnosperm, 4 megaspores are formed. In these 4 megaspores, as in angiosperms, 3 degenerates, 1 functional megaspore. 1 functional megaspore. This megaspore undergoes free nuclear division and produce many haploid nuclei. Undergo free nuclear divisions and produce many haploid nuclei. Later these nuclei get surrounded by cytoplasm to form a haploid tissue called female gametophyte. As female gametophyte provide food for developing embryo, it is also called endosperm. So endosperm or female gametophyte in gymnosperms is always haploid. So 4 megaspores, this in 3 disintegrate. One is functional, which undergoes free nuclear divisions. Many haploid nuclei are formed. All the haploid nuclei later get surrounded by cytoplasm to form a special tissue called female gametophyte or endosperm. So, endosperm of gymnosperms is in haploid. Why it is haploid? Because it is a pre-fertilized product. Female gametophytic tissue formed from haploid megaspore functions like endosperm. So, here assertion and reasoning are one related to each other. So, the answer will be both A and R are correct. R is the correct explanation of A. Non-archegoniates in which siphonogamous type of oogamy is present. Non-archegoniates, that means archegonia is absent. And siphonogamous type of oogamy. Siphonogamous type of oogamy and non-archegote will be angiosperms. So, in these angiosperms, primitive land plants are not angiosperms. Vascular cryptogams are pteridophytes. Fruitless spermatophytes are gymnosperms. Gymnosperms are archegoniates. They are not non-archegoniates. So, fruit-bearing spermatophytes. Fruit-bearing spermatophyte is angiosperm. So, non-archegoniates in which siphonogamous type of oogamy. What is siphonogamy? Movement of male gametes from one place to other place with the help of a pollen tube. So, siphonogamous type of oogamy present is fruit-bearing spermatophytes. Fruit-bearing spermatophytes means angiosperms. So, answer is 4. Assertion reasoning. Conjugation tubes are not formed in the sexual reproduction of Spirogyra jogensis. Spirogyra jogensis is at attached form, sedentary form, attached to rocks in a pond. This Spirogyra jogensis, whenever direct lateral conjugation occurs, first the cells above the holdfast female cell and above the female cell haploid, that is male cell, are present. So, only 2 cells involved in the process, and in the male cells eplenogamete is formed, in the female cell eplenogamete is formed, but eplenogamete of male cell shows a small projection called as conical process. This conical process pierces through the septa almost at the center, ultimately enters the other cell, fuses with the eplenogamete there, resulting in the formation of zygospore. So, here there is no question of formation of conjugation tube. So, conjugation tubes are not formed in the sexual reproduction of Spirogyra jogensis is correct. Both lateral and scalariform conjugations are found in Spirogyra jogensis is wrong. Spirogyra jogensis shows only direct lateral conjugation. Lateral and scalariform conjugations are found in Spirogyra graciana. So, here conjugation tubes are not formed in the sexual reproduction of Spirogyra jogensis is correct, but Spirogyra jogensis does not show lateral and scalariform conjugations is wrong. So, A is true and R is false. A is true and R is false. 3 will be the option. Which of the following true with regard to gymnosperms? So, we have discussed about thallophytes, bryophytes, pteridophytes, now gymnosperms. Gymnosperms show direct pollination. What is direct pollination? The 3-celled microspores liberated from the microsporangium with tube cell, generative cell, prothallel cell directly comes and attaches to the ovule. No question of going to stigma, then stylar canal, then to ovule. Here the pollen grains or microspores directly reach the ovule. So, definitely direct pollination. Vessels absent in xylem, companion cells absent in phloem. Definitely xylem vessels are absent in xylem, companion cells are absent in phloem, and they are replaced by albuminous cells. Ovules contain archegonia. So, definitely each ovule in cycas contains 2 to 6 or 2 to 8 archegonia. Sporophylls are aggregated as strobili. Sporophylls means spore bearing leaves. They are aggregated to form cones or strobili. Male cone is very much present in cycas. So, which of the following is true in regarding gymnosperms is all the 4. 1, 2, 3, 4. So, answer will be 3. This is a matching regarding Spirogyra species. Single chloroplast per cell, eplanospores, both calariform and lateral conjugation, filament with polarity. Single chloroplast per cell is Spirogyra venkatramani. So, A5. Eplanospore, Spirogyra eplanospora, B3. Both calariform and lateral conjugation, Spirogyra graciana, C4. Filament with polarity, Spirogyra jogensis, 1. So, answer is A5, B3, C4, D1. So, single chloroplast per cell is Spirogyra venkatramani. Eplanospores are Spirogyra eplanospora. Farlovi is echinids. Farlovi means Spirogyra. Farlovi always produce echinids. Then both calariform and lateral conjugations are found in means Spirogyra graciana. And filament with polarity. Filament with polarity means majority of the free floating filament in Spirogyra does not show an apical cell and basal cell. But fixed floating filaments like Spirogyra jogensis, Spirogyra adnata, the basal cell is modified into a colorless special non-green root-like fixing cell called holdfast or hapteron. The apical cell is clearly mentioned. So, apical cell, basal cell differentiation will be there. That is called polarity in Spirogyra jogensis. So, answer is 1, A5, B3, C4, D1. Assertion reasoning. Only algae and fungi are placed in thallophytes. Only algae and fungi are placed in thallophyta is correct. Autotrophic thallophytes are algae, heterotrophic thallophytes are fungi. Flowers are absent in algae and fungi. It is also correct. Flowers are absent in algae and fungi because they are all belonging to cryptogams. But because the flowers are absent in algae and fungi, they are not kept in thallophyta. So, A and R are correct. R is not the correct explanation of A. So, only algae and fungi are placed in thallophyta is correct. Only algae and fungi placed in thallophyta is correct. Flowers are absent in algae and fungi is also correct because they belongs to cryptogams, not phanerogams. So, but because the flowers are absent in algae and fungi, they are not kept in thallophyta. Because of so many other reasons, they are kept in thallophyta. So, A and R are correct. R is not the correct explanation of A. Second will be the answer for this. Now, in a Spirogyra, if 20 cells are involved in isogamous calariform conjugation, what would be the number of gametes, zygospores and empty cells? Isogamous conjugation means definitely both the male gametes will fuse. 20 cells are involved in the isogamous calariform conjugation. So, 20 gametes are formed, 10 zygospores are formed, and empty cells will be 20 because on both sides 10 cells become empty. So, if Spirogyra, in a Spirogyra, if 20 cells are involved in isogamous calariform conjugation, isogamous means the gametes of both the filaments move and ultimately zygospores are formed in conjugation canal. So, what would be the number of gametes means 20. Both sides gametes are formed. What would be the number of zygospore means 10 because two gametes fuses to form one zygospore. And what would be the number of empty cells means all the 20 cells because total aplanogamete moves and fuses in conjugation canal to form zygospore. So, answer is 20, 10 and 20. 20 gametes, 10 zygospores and 20 empty cells. Arrange the following plant groups in the order of primitive cryptogams, most advanced archegoniates, most advanced cryptogam and first embryophytes respectively. Primitive cryptogams will be fourth one, thallophyta. These are primitive cryptogams. Here answer is 3. Then second is primitive cryptogam is thallophyta. Most advanced archegoniates are gymnosperms. Most advanced archegoniates are gymnosperms. Most advanced cryptogams are pteridophytes. Most advanced cryptogams because these are vascular cryptogams. Then first embryophytes are bryophytes. So, answer is 4, 2, 3, 1. I will repeat this question. Arrange the following plant groups in order of primitive cryptogam. Primitive cryptogams means thallophyta. Most advanced archegoniates means gymnosperms because even though they produce archegonia, they are definitely most advanced, possessing many characters related to angiosperms. Most advanced cryptogams by the development of xylem and phloem, vascular cryptogams will be the most advanced cryptogams. Example that is in this is pteridophytes and first embryophytes. For the first time in plant kingdom, zygote starts mitosis only in bryophyta. So, definitely bryophytes show embryo. So, answer is primitive cryptogams are thallophytes, most advanced archegoniates are gymnosperms, most advanced cryptogams are pteridophytes and first embryophytes are bryophytes. 4, 2, 3, 1 is the answer. This is an assertion reasoning. In Spirogyra aplanospora, much genetic variations does not occur. In Spirogyra aplanospora, much genetic variations does not occur because here fusion of gametes does not take place. Just cell contents condenses to form a special non-motile thick-walled spore called aplanospore. Every cell in Spirogyra aplanospora produces aplanospore. Spirogyra aplanospora multiplies exclusively by aplanospores. Because it multiplies exclusively by aplanospores, there is no genetic variation here. So both A and R are correct. R is the correct explanation of A. First option will be for this. Study the following table and identify the incorrect combination. First tracheophytes or first archegoniids, pteridophytes wrong. First tracheophytes is correct, but first archegoniids are bryophytes. Then first embryophytes, first archegoniids, bryophytes, correct. First embryophytes are first archegoniids and they are bryophytes is correct. Last archegoniids, first seeded plants, gymnosperms is correct. Because for the last time archegonia is formed in gymnosperms, in angiosperms, no question of formation of archegonia. So last archegoniids, first seeded plant. For the first time a seed-like structure or seed is formed in gymnosperm. Last cryptogams, first true land plants, pteridophytes. Pteridophytes are definitely last cryptogams. Thallophyta, bryophyta, pteridophyta. These are first true land plants with the development of vascular tissue system. Successfully they started growing on the land, and pteridophyta is correct. So study the following and identify the incorrect combination is first one. First tracheophytes are not first archegoniids. First tracheophytes are pteridophytes. First archegoniids are bryophytes. So first option is wrong here. Identify the incorrect statement regarding Spirogyra jogensis. Conjugation tube is absent in its lateral conjugation. It is correct because directly this aplanogametes pierces through the septa and enters the other cell. Male gamete consists of conical process. Definitely male gamete consists of conical process. With that conical process only it pierces through the septa and enters the other cell. Only 2 zygospores are formed in each filament. This is wrong. Always only 1 zygospore is formed in each filament. Ratio between male gametophyte and female gametophyte is 1:1. Male gametophyte is filament, female gametophyte is zygospore or any other thing. It is definitely only 1 male gamete and 1 female gamete is formed, so 1:1. So identify the incorrect statement regarding Spirogyra jogensis is 2 zygospores formed in each filament is wrong. In any filament only 1 zygospore is formed. So third option. When 2 spirogyra filaments of 8 cells each are undergoing isogamous calciform conjugation. Isogamous, previously I told, both the aplanogametes move. Conjugation, the total number of gametangia, empty cells and zygospores. Total number of gametangia will be 16 because 8 plus 8. Total number of empty cells, all the 16 will become empty. 8 this side, 8 that side become empty. Zygospores formed will be 8. So 16 cells. 16 total number of gametangia. This side one aplanogamete, that side one aplanogamete, correct. Empty cells are 16. Both 8 plus 8 become 16 and zygospores will half 8. So answer is 16, 16 and 18. This is an assertion reasoning question. Heterotrophic thallophytes show progressive reduction in sexuality. Heterotrophic thallophytes means fungi. They definitely show progressive reduction in progressive evolution in sexuality is found in algae, autotrophic thallophytes. Lower group of plants have low sexual reproduction, higher group of plants have advanced sexual reproduction. Whereas in the case of fungi, lower group of plants have much sexual reproduction, higher group of plants do not have. For example, last class in fungi, that is Deuteromystes, they are also called fungi imperfectae because sexual reproduction is absent. So heterotrophic thallophytes show progressive reduction in sexuality is correct. In evolved fungi, sex organs are undeveloped. Because they show progressive reduction in sexuality, in evolved fungi, that means advanced fungi, sexuality, sex organs are undeveloped. So A and R are correct. R is the correct explanation of A. The most evolved sexual reproduction method in Spirogyra. The most evolved sexual reproduction in Spirogyra is calciform conjugation. The most evolved sexual reproduction in Spirogyra is calciform conjugation. So isogamous calciform conjugation is wrong because both the male gametes or both the aplanogametes move. Anisogamous monoecious conjugation is wrong. Physiologically anisogamous lateral conjugation is wrong. Even lateral conjugation is also physiological anisogamy because both the male gametes or both the gametes are structurally similar but functionally dissimilar. Physiologically anisogamous dioecious combination, dioecious conjugation, that is Scalariform conjugation is very, very true. So, the most advanced evolved type of sexual reproduction is physiologically anisogamous dioecious conjugation. Answer is 2. Stele is first formed in. Stele means the place with xylem and phloem is called stele. Gametophyte of pteridophytes is wrong. Pteridophyte, gametophyte is either prothallus or any other structure which is completely made up of chlorenchymatous tissue. No question of stele is formed. Sporophyte of bryophyte. Sporophyte of bryophyte means foot, seta and capsule. Their central cylinder will be there. No question of stele. Sporophyte of gymnosperms. Sporophyte of gymnosperms contains stele but not first form. So, first formed stele. Stele is first formed in sporophyte of pteridophytes. That's why pteridophytes are very popularly called vascular cryptogams. Third option. The correct sequence of nuclear condition through which the zygospore of Spirogyra passes from the time of its formation to its germination. First, it is monokaryotic because zygospore is monokaryotic. This is zygospore with 3 layers around it. This is monokaryotic. That means it shows one diploid nucleus. It undergoes reduction division and produces a temporary tetranucleate stage. That means 4 haploid nuclei are formed. So, temporary tetranucleate stage. 3 disintegrate and 1 remain functional. 3 disintegrate and 1 remain functional. Again it becomes monokaryotic. So, correct sequence of nuclear condition in the germinating zygospore of Spirogyra is first monokaryotic, again tetrakaryotic, again monokaryotic because out of 4, 3 disintegrate, 1 remain functional. So, monokaryotic, temporary tetranucleate and monokaryotic is correct sequence. So, the correct sequence of nuclear condition through which the zygospore of Spirogyra passes from time of its formation to its germination is monokaryotic first, temporary tetranucleate next, monokaryotic next. So, third is the option. Arrange the following events in a sequence with reference to the scalariform conjugation in Spirogyra. Pairing of conjugating filaments, formation of zygospore, development of papillae between female and male cells, formation of conjugation tube, migration of male gamete into the female cell. So, first is pairing of conjugation filament. Fourth is the option. Pairing of conjugating filament is first. Then development of papillae between female and male cell is second. So, 1 first, 3 next. Then 4, migration of 4, formation of conjugation tube. Once both conjugating filament come close, papillate outgrowth come in contact with each other, the wall at the point of contact dissolved by cytase enzyme resulting in the formation of conjugation tube. So, 4 will come. Then 5th one, migration of male gametes into the female cell. Migration of male gamete, physiological anisogamy takes place. And finally, formation of zygospores. So, what is the correct sequence? First, pairing of conjugating filament. Next, formation or development of papillae between male and female cells. Then, formation of conjugation tube. Then, migration of male gamete into female cell. Now, finally, formation of zygospore. So, the correct sequence is 1, 3, 4, 5, 2. Which of the following is parietally located in the cells of Spirogyra? Parietally means outwards. Cytoplasm and chloroplast. Cytoplasm and nucleus. Nucleus is present almost at the center attached by cytoplasmic strands. Nucleus and vacuole. Nucleus is present at the center, vacuole is present at the center but very large. Cytoplasm and vacuole. Vacuole at the center, cytoplasm at the periphery. So, which of the following are parietally located in a cell of Spirogyra means cytoplasm and chloroplast. Why? Because of the presence of central large vacuole, primordial utricle is formed. So, cytoplasm and chloroplast will be the answer for this. First option. Identify the correct statement about the chloroplasts of Spirogyra. They run parallel to the central axis or cell axis is correct. They are spirally twisted and anti-clockwise. Spirally twisted and anti-clockwise is correct. They are parietally located in the cell. It is also correct. So all is correct. They run parallel to the central axis or cell axis. They are spirally twisted and anti-clockwise is correct. They are parietally located in the cell means they are peripherally located in the cell is correct. So all of this is the answer. Fourth is the answer for this. Shrinkage and rounding up of the cell protoplast occurs during the formation of eplanospores, during the formation of echinids, total cell wall and protoplasm involves. Gametes are formed only by the fusion. So answer for this will be 4. Shrinkage and rounding up of cell protoplast occurs during the formation of eplanospores and gametes. So eplanospores, and it's not 1, it is 4. Eplanospore and gametes 1 and 3 is the answer for this. Which of the following is a thick walled vegetative resting structure? Asexual reproduction in Spirogyra is by 2 methods. One is echinid, the other is eplanospore. Echinids are formed in Spirogyra farlovi. Eplanospores are formed in Spirogyra eplanospora. During the formation of what you call echinid, the total cell involves here and the total cell wall protoplasm forms what you call echinid. Whereas in the case of eplanospora, only protoplast condenses to form a special gamete-like or spore-like structure called eplanospore. So which of the following is a thick-walled vegetative resting structure means echinid. Echinid is thick-walled vegetative resting structure. Eplanospore is also thick-walled, but total cell is not involved. Zygospore is not a vegetative, it is a reproductive structure. Azygospores are formed by non-functional eplanogametes. So answer is echinid. Arrange the following events of sexual reproduction of Spirogyra in correct sequence. 1, 4, 3, 2. What is 1, 4, 3, 2? Male papillae formation is first. Then strain papillae formation, that means female strain also starts producing formation. Then cytase action occurs immediately. Then movement of plus strain gamete. So male papillae formation occurs first, female strain papillae formation occurs next. Now both the papillae come in contact with each other by the action of cytase, definitely wall dissolves and conjugation tubes are formed. Once the conjugation tube is formed, movement of strain gamete, plus strain gamete moves from plus filament to minus filament. So answer 1, 4, 3, 2, first option. Asexually, Spirogyra reproduces by. Asexually, Spirogyra reproduces not by fragmentation, because fragmentation is vegetative propagation, where by the gelatinization of middle lamellum, by the continuous movements of small water animals, and by the maximum effect of water currents, the total filament in Spirogyra breaks up into bits. Each bit is called fragment, and each fragment produces complete filament. So it is vegetative propagation. Coming to asexual reproduction, it is by echinids and eplanospores. So answer is 1 and 2. Scalariform conjugation is isogamous, anisogamous, physiological, anisogamous. It will be isogamous. Isogamous means both will fuse. It is not anisogamous because both the male gametes are almost, both the male and female gametes, eplanogametes are almost similar. Physiological anisogamous is correct. So answer is isogamous and physiologically anisogamous, fourth option. Now this is a matching tabular form. Spirogyra affinis, Spirogyra jogensis, Spirogyra graciana, Spirogyra farlovi. Spirogyra affinis, indirect lateral conjugation, third option. Spirogyra jogensis, direct lateral conjugation, fourth option. Spirogyra graciana, scalariform conjugation, fifth option, because graciana shows both scalariform and lateral conjugation. Spirogyra farlovi, echinids, second option. So A3, B4, C5, D2, answer is first option. Spirogyra affinis, indirect lateral conjugation. Spirogyra jogensis, direct lateral conjugation. Spirogyra graciana, scalariform conjugation. Spirogyra farlovi, echinate formation. So first option A3, B4, C5, D2. Similarity between Spirogyra and Rhizopus that they both have. Similarity between Rhizopus and Spirogyra. Spirogyra is an algal member, autotrophic thallophyte. Rhizopus is a fungi, heterotrophic thallophyte. Uninucleate gametangia is wrong because in Rhizopus multinucleate. Cenocytic eplanospores is wrong. Again in Rhizopus it is cenocytic. Uninucleate zygospores is wrong because uninucleate zygospore is formed in Spirogyra. Multinucleate zygospores are formed in Rhizopus. What is the similarity? Is thick-walled diploid zygospores. In Spirogyra, 3-layered wall is present. Outer exospore are exine, middle mesospore are mesyne, inner endospore are intine. Whereas in Rhizopus, 2-layered thick wall is formed. Outer exospore and inner endospore. So, similarity between Spirogyra and Rhizopus is that both have thick-walled diploid zygospores. Fourth option. The number of germ spores formed in each germ sporangium in Rhizopus. The number of germ spores formed in each sporangium. Suppose the plus mycelium has 40 nuclei. Think that. Minus mycelium has 40 nuclei. When both these fuse, 40 diploid nuclei are formed because both the haploid nuclei fuses to form the same number of diploid nuclei. These 40 diploid nuclei undergoes reduction, division and form 160 meiospores or germ spores. So 40, 160. That means the answer here will be equal to that of diploid nuclei of zygospore is wrong. Half to that of diploid nuclei in zygospore is wrong. 4 times to that of diploid nuclei of zygospore is correct. 4 times to that of haploid nuclei of zygospore is wrong. So the correct answer is 4 times to that of diploid nuclei of zygospore. Why? Because after the formation of zygospore by reduction division, the diploid nuclei undergo reduction division and produce 4 times to that of diploid nuclei in zygospore. So answer is 4 times to that of diploid nuclei of zygospore. So for 30th question, the number of germ spores formed in each germ sporangium of Rhizopus is always 4 times to that of diploid nuclei of zygospores. During the period of dormancy, the zygospore of Rhizopus contains haploid nuclei and plus and minus 10 because of karyogamy is not yet completed is wrong. Diploid nuclei resulted by karyogamy is correct. Haploid nuclei of plus and minus 10 resulted by the reduction division of following is wrong. Only one diploid nucleus because the other non-functional disintegrate is wrong. So answer is diploid nuclei resulted by karyogamy is the correct answer. Second option. Find out the correct sequence of events in the sexual reproduction of Rhizopus. Plasmogamy, karyogamy, nuclear pairing, meiosis is wrong. Nuclear pairing, plasmogamy, meiosis, karyogamy is wrong. Karyogamy, meiosis, nuclear pairing, plasmogamy is wrong. The correct option is first fusion of protoplasm, plasmogamy. Then nuclear pairing, equal number of nuclei fuse, the unpaired nucleus disintegrates. So nuclear pairing. Then karyogamy. Karyogamy means once nuclear pairing, immediately karyogamy takes place. Then meiosis. Then meiosis takes place. Meiospores are formed. Second is the correct option. Arrange the following events of sexual reproduction of Rhizopus in a sequence. Arrange the following events of sexual reproduction of Rhizopus in a correct sequence. First, promycelium. First is zygophore formation. What is zygophore? First, a vegetative mycelium plus and minus identified. But they are called vegetative mycelia which cannot involve in sexual reproduction. These vegetative mycelia, in the presence of a special sex hormone or chemical substance called trisporic acid, converts into some type of hyphae which can involve in sexual reproduction, and these are popularly called zygophores. So zygophores are vegetative mycelia. Vegetative mycelia cannot perform sexual reproduction. Zygophores perform sexual reproduction. So zygophores. So first option, zygophore formation. So 4 is zygophore formation. Next one, Zygospore formation. Once the zygospores are formed, nuclear fusion takes place and ultimately fusion results in zygospore form. Then promycelium formation. The zygospore gives rise to a special outgrowth called promycelium, and finally germ spore formation. So first is zygospore formation, fourth. Next is zygospore formation, second. Then is promycelium formation, 3. And last is germ spore formation, 2. So 4, 1, 3, 2 is the correct sequence in the sexual reproduction of Rhizopus. Arrange the following events of a sexual reproduction in Rhizopus in sequence. First, sporangiophore growth, then sporangial development, then cleft formation, next cleavage. So first is sporangiophore growth, second option. Then 1, sporangial development. Then cleft formation. Cleft formation results in the formation of columella. And finally cleavage. So first option for this 2, 1, 4, 3. Arrange the following structures of Rhizopus with increase in their number of nuclei. First sporangio, I don't say sporangiophore, answer is 3. First initial stage of germ spore, because it is haploid, only 1 nucleus. Then sporangiospore, 2 to 8 nuclei. Then chlamydospore, all the cell aggregate to form surrounded by thick cuticle chlamydospore. And stolon. Stolon has maximum nuclei. So correct option is initial stage of germ spore, sporangiospore, chlamydospore and stolon based on the number of nuclei present in them. Third option. Now it is a matching. Match the following and choose the correct option. Zygophore, sporangiophore, rhizoids, stolons, formed from zygospore, hyphae for digestion, more stouter hyphae, has copulating branch, collimated hyphae. This is A is 4, has copulated branches because zygophore form copulated branch. B5, sporangiophore is collimated hyphae. That means sporangiophore definitely contains columella. Zygosporangium does not contain columella. Rhizoids. Rhizoids are hyphae for digestion. They secrete exoenzymes which acts on medium and converts it into food material. Stolons, stouter hyphae present in the structure, more stouter hyphae. So A4, B5, C2, D3. Next. Match the following and choose the correct match. Structure of Rhizopus formed from promycelium, sporangiophore, progametangium, mycelium. Prothallus, zygospore, spore, stolon, zygophore. For this A2, promycelium comes from zygospore. B4, sporangiophore comes at the base or from the stolon. C5, progametangium comes from zygophore. And mycelium comes from prothallus. So mycelium is regarded as prothallus. I don't say prothallus, it is 3. Mycelium comes from spore. So A4, A2, B4, C5, D3. Prothallus. Mycelium always comes after the germination of spore. This is a 3-column tabular form. Rhizoids, vegetative and digestion. Correct. Zygophores, reproductive production of gametangia. Correct. Sporangiophores, reproductive production of germ spores is wrong. Promycelium, reproductive production of chlamydospore is wrong. Promycelium produces only germ sporangium. It does not produce chlamydospore. So 1 and 2 are correct, 3 and 4 are wrong. Next. This is another column. Type of spores, nature of wall, type of reproduction involved. Eplanospore, thin-walled, asexual, correct. Zygospore, thick-walled, sexual, correct. Germ spore, thick-walled, sexual, correct. Chlamydospore, thin-walled, asexual is wrong. So germ spore, thick-walled, sexual is also wrong. So answer is for this is eplanospore, thin-walled, asexual is correct. Zygospore, thick-walled, sexual is correct. So 1 and 2 are correct. Third option is the correct for this, and all the others are wrong. Rhizopus sexualis are homothallic species. Rhizopus sexualis is a homothallic species, correct. The gametangia of 2 hyphae of same mycelium are compatible in sexualis. Compatible means each mycelium can complete the complete reproduction. So definitely compatible. So Rhizopus sexualis are homothallic species, correct. The gametangia of 2 hyphae are of the same mycelium are compatible of Rhizopus sexualis is correct. So definitely both A and R are true and R explains A, first option.